G'day. Excuse me. Woohoo. Hope you're well. I was just watching that video. That's a cool video, that breeding video. If you haven't seen it, go watch it. I just watched that again and I didn't really get started. Sorry. I'll have to load everything up. Wait for people to come and say hello. Hello. G'day, Mr. Nexus. How are you? What am I gonna? I'm gonna share some slides. We're going to talk about some cannabis. Thanks, Nexus. Yeah, it's a good video, mate. We can talk about it soon. If you've got any questions, that's what I thought we'd do today, because I'm not really going to show any slides. Well, last week went pretty good just answering questions, and I'd rather help people out than choose for what used to watch. So if you've got any questions, that would be good. So I can show you this week's latest research stuff and some technology things that I found in cannabis. And then questions again, because yeah, I'd rather help people. So keep that in mind. All right, where's my? I'm going to share the slides and start. Here we go. So I'll read. I'm not going to be able to see the chat. So hello. I see 4 people that have joined. Thank you and hello. G'day, Captain Calmag and Lene. Good morning to you guys. I'll run through some stuff, and then we'll get to some questions, Captain Calmag. There's only a few things I'm going to show and just talk about, but they're interesting. There's this week's stuff that I found in cannabis that I thought is pretty cool. All right, share screen, entire screen. All right, I think it's good. This one. These are the things here. So the first one's on, oh yeah, cool, not really interesting, but it's just to show the progression of how cannabis is going. And this is in California, so it shows you from the 3 years in the middle here with the cursor, it's 2020, 21, 22. So the progression of the years, these are the sales that they've done on the different amounts of quality for the weed. And it's how it's gone down, progressively gone down. And they put a new exotic range in for all these new exotic strains, and that sort of stayed up pretty good, but everything else is on a sliding scale. So can't remember the point was that, but, and down the bottom, what does it show down here? That the price difference from the previous years by how it's gone up, and that's kinds of what I was talking about at the top. This is I thought it's pretty cool because it more defines it about where you're going to find your deficiencies and why you should find the deficiencies in the certain leaves on the certain layers. It's because to do with mobile and immobile elements. So if the leaves on the lower bits, that's where you're going to see all your mobile element deficiencies, and the immobile ones, because elements can't travel around, you're going to see them on the new shoots that come out from the top ones, which is on the upper. So that's your calcium, your boron, magnesium, and now your 3 immobile. And then you've got your, what do they call it? Variable mobile ones. Now, on that new list that I've just found out from this year's notes, that there's a variable mobile, and that has the iron, copper. I don't know about the silicon and manganese, I can't remember. But anyway, I can definitely confirm these 3 are. But that's, yeah, kind of. And this is down here, pretty poor examples, I'm sorry, but with phosphorus, it goes dark purple. It goes like this shade over here, it's green. It'll go like a darker purple. That's a horrible example. And the nitrogen, that's like, it's not the best either. That's sort of at a very, very, I don't know, it's the veins in the middle go green. And that's an easier way to depict it. This is probably in really extra late stages when it's all extra yellow, but you want to pick them up at the start, not when they're all bloody yellow. So it goes, the greens, the veins in the middle go green. Where's if someone asks me later, I'll pull up a photo. I've got some good better photos of things like that. And then even this one for potassium, on the edges, your leaves crisp up. So this might be an early stage of potassium deficiency. So they go brown, and that's a good difference between magnesium, because the magnesium has an overall blend and it won't die off on the end, because magnesium's involved in your chlorophyll in the middle of the atom. It's got magnesium in the center, and then its anions branch off, and that's where its N minuses on it. Yeah, cannabis chocolate. I'm a chocolate fiend as well as a cannabis fiend. So this has got 2 words. Oh, look, it's CC, cannabis chocolate, AussyCC, cannabis chocolate. Oh, yeah? Oh, I hope you're laughing too. Jesus, but I just saw it. I thought, wow, that's really nice because it looks like it'd be a nice blend or something, not just thrown in boxes and done normal, not those wraps and things like people normally do. And this is progression in agriculture. This is hydrogen powered. How awesome is that? It's water. So water powers this friggin' tractor. Oh, I love it. And the reason why this didn't happen, back in the turn of the century, they were making machines like this, but I think it was something to do with the big Henry Ford and those types of big companies bought them out, and they squashed it because they wanted to run their fuels and shit through it. So this is just, I just love it. Oh, here's a mouthful. All right, where's the drink? So Gregor Mendel, he's the legend of breeding. So there's Mendel's laws that's been written about him, and from here all of his breeding. A lot of the course that I did, genetics and genomics, the 13-week course, was pretty much a shitload was just on him, because he's just done so much research and stuff in the mid 1800s on breeding. And the main 7 things that he did was the flower color, the length, the position, and then there was heaps of seed stuff to do with the seed color, shape, the pod color, and the pod shape. What can I tell you about this? That's the law of dominance. I suppose I'll read it then. That's the law of dominance down there. When an organism has 2 different alleles from one trait, one allele will be dominant over the other. The allele is a gene in like a chromosome. You've got a chromosome leg, the long thing, and then there's all types of genes in it, and then the allele is in the gene, so you can have different forms of the gene and their alleles. Excuse me. Of the offspring. All of the offspring had purple flowers. Mendel allowed the purple flowers to self-pollinate, and then they got 1 out of 4. So if you're going to do a breed, cool. This is how you do a test. So if you lay out 4 seeds, and from the 4 seeds, if you were to get 3:1, like this, if you're going to get this ratio, if you put 4 seeds out, that means you've got a dominant gene in purple. And then if you've got a 1 would be the recessive gene. So you can tell instantly what traits you're getting from laying 4 seeds out to which genes you want to work with in your breed. Because then you'll know, all right, if it's a dominant gene, piss easy. There's a high chance I'm going to transfer the alleles or the genes I want across when I'm going to do a breed. And if it's a recessive trait like this one, you're going to really have to work on it to transfer that gene across. Like with my who cares about my stuff? I'll just explain this stuff. This is pretty snazzy. So this is due to with the transcription factors in cannabis with the mixed TA gene, and when they used it in tobacco, they found that it overexpressed trichomes in tobacco, and it was a really cool thing that they're just starting to play with the codons and the genes in different varieties now. So like that, because liverwort, that's got THC in it, so it'd be good to muck around with that. Because remember back in the day, Congo was the highest percent in one of my books. In the mid 80s, it was 6%. It was testing, and liverworts, I think it's like around 1 or 1%. So potential. Oh yeah, this is today. Deficiencies, breeding, silicon, and some CBD. Oh yeah, here we go. The silicon stuff. It's a mouthful too. So over here, A. This is silicon. How silicon helps, or silica helps in the plant with us, our cannabis plants. So it's an osmoprotectant. That helps with drying out. So it's beneficial for the plant to have the silica, because if it's going through a dry phase, it helps with by producing the proline, glycine, betaine, which are osmoprotectants, which react and they're turned on and help when plants get really dry. So in general, it helps with stress and what is it to do with bacterial and fungi as well? It helps with them. So if you're growing with living beneficials, which you should be. Well, unless you're growing in a sterile environment in a lab, you're not. But they really help. Can't remember what I was getting to there. All right, back to this. C, well, they're the enzymes, catalase, superoxide dismutase. That helps with them. So it helps with initiating those, which are antioxidants. So you want to really reduce them because the antioxidants are bad. They're free radicals. So that's the whole thing for them. It produces stress proteins, produces Li, which is a stress protein, and reduces and helps with it. The polypeptides, that's the aminos, so it helps with them, and they're all precursors, for good functions. Cytochrome complex B, that's your what's it? Cytochrome and phytochrome. So cytochrome's up the top, so that's the blue. So that produces all your blue spectrum in your photosynthesis, modulates phytohormones synthesis. Oh yeah, it helps regulate and modulate your hormones in your plants, and it gives an overexpression of the SAMDC gene, which is a polyamine synthesis, which helps in that helps with your hormones as well, and it promotes membrane stability, increases the cell walls, and this was what's this? Silicon improves cell extensibility in growth regions in roots. Extensibility, cell elongation. It helps with elongations of the cell roots. Now, there's the cell wall over there. So there's some good things that it is beneficial in it, because in those 19 essential elements for plants, they were saying that silica was a variable element, because some plants it's not necessary, but cannabis it is. Oh, look at this. Isn't that sexy, eh? It's just so nice. And look, it's aged. That's the color you get with amber trikes when they're aged. They look that sort of amber. I wonder if it's got a phosphorus deficiency. But nice trike. Zoomed in. Last week I showed the one that was mega zoomed in. I thought, wow, this one's not quite zoomed in, but you can still see all the cell walls in it. Look at that. That's cool. With all the lignin, the cellulose, and hemicellulose protects it. All the different types of phytols and chemicals and flavonoids inside. This is to show, this is cool. This is to show the effects of the heat and the sun's radiation that comes down. So it's only going to get worse. So if you grow outdoors, think about this. If you've got very hot summer days, like now it is in North America and Northern Hemisphere, you've got this happening. So 37, that's 100 F outside. That's in the air temperature. So the shade temperature would be 100 F. And then in the sun, look on your car, you've got, what's that, 150, 160 F. It's just really, really high. So with the canopy over the top, you're dropping it heaps of degrees. You can see that. Well, what's 27 gone back to? 27 Celsius. Is that 60? Nice. 80. 80 F. So it's dropped down from 100 to 80. So really nice. But you can do outside. You can keep that in mind. If you're getting a really hot day, you can go, all right, well, look, the effect of the direct sun rays is going to be very, very hot. Well, can you shade it? Can you put some cloth up? Can you raise the humidity? Because temperature is in direct correlation with the humidity. That's why it's called relative humidity. It's relative to the temperature. So if you can raise that humidity, like by putting a sprinkler around or something, this temperature will drop even in the actual, like here. Hemp seeds. Oh yeah, this is rad. This is really cool. We're all taught about flax seeds and how they're really healthy growing up. Well, look at the difference. It turns out hemp seeds overpower it. How cool is that? So if you're breeding, and your seeds go, if you don't store them correctly, like you should, below 4 degrees and in the back of the fridge, or really low humidity, they'll expire after a couple of years. So don't get rid of them. They'll still have these good benefits in it. So eat them. And look at the difference. So I'm going to go through this because this is really, really cool. So omega 3 to 6, the difference is a 1 to 10 ratio in flaxseed. Hemp is 1 to 3. The protein is twice the amount in hemp. Potassium 800, 1200 in hemp. heaps difference. Magnesium, double. Zinc, double. Iron, nearly. And manganese, triple. So that's per 100 grams. So that's what? 3 and a bit ounces of seed. And they've done this. These are the numbers that you get out of that. 112 grams is 4 ounces. So that's awesome. I was at the end. Oh, it's the end already. Really? Oh, that's the end. Sorry. Not sorry, but that's the best I can do. Shit. Looks like we're going to be on questions. I'm going to be scrolling up and looking for questions because I'm not going to go through my notes because it went pretty good last week. How I was just answering a few questions, so I'm going to scroll up and see if there's any. And say g'day to the people, the other people, my green ones I didn't say g'day to before. And Aussie Autos. That's right, there's one right at the top. Yeah, Captain Calmag, thoughts on using rice flour. Actually, hang on. Before I get to that question, there was one more thing. Yeah, this thing. It's just to do, it's small, it's nothing special, but it's to do with the legalization of cannabis. Where do I get to share the screen? And the progression. Now it's going good, and now Australia needs to progress. So Argentine Supreme Court rules that you can grow your own medical cannabis at home now. Finally, it says of July 19, 2022. So that's really cool. And there was, I think Spain was in the guts of nearly getting it all officially done too. And Australia is just, again, going to be the last on the planet to allow people to grow at their home. Just so, yeah, they reckon, yeah, we want to stay ahead of the times, one of the best in the world. Good on you, Australia. Pig's arms. All right, back to the questions. Thoughts on using rice flour for pollen storage? Oh, Captain Calamang, rice flour. Rice flour. Well, that's a desiccant, and it's going to dry it out. So I suppose I wouldn't directly store it in it because you won't be able to get it out and it'd contaminate it, and you don't want rice flour in your plants to block up all your pores. But I store mine in clippy bags, and those clippy bags have paper towel in it. And then I put the clippy bags inside a container, and that container has rice in the bottom of it, uncooked rice. So if any moisture was to get in that, the rice would absorb it before it got into the side of the clippy bag. And then if anything got in the clippy bag, if the rice was full for some reason, that the moisture in the paper towel would stop it. And I've done that for quite a few years, and that seems to work all right. I hope that helped with your rice flour pollen storage question, mate. Update me if it didn't. Cheers. G'day, Cherub Bro. Cheers, everyone. Just reading the questions, if there's any reading down the chat. Did you increase dosage at all or stayed with the 2 leaves? Green Lung says. Uh-oh. I'm not sure. Kay also has. I don't know what it means, Green Lungs. Did you increase dosage at all or just stayed with the 2 leaves? Don't know what I'm talking about, mate. Sorry. Awesome. I learnt my Pineapple Express last year. Thought was a potassium deficiency, then worked out was still in veg with nitrogen toxicity. Yes. Well done, Aussie Autos. There's so fine line with deficiency and toxicity. It really, truly is. And that's why people say go on the lighter side before, if anything, because it's hard. Sometimes they can go like a toxicity, you can have a hidden hunger even, which means that the plant is not giving the yield it should, and it's not showing any deficiency signs. So that's even a full-on one that's a diagnosis needs with. So good on your Aussie Autos. Next is a true mad scientist. Yes, he is. But he was talking about Mendel. Yes. Gregor Mendel's rad. All these different experiments, so helpful for the industry. Green lungs, there's still very low on level, which is, mate, in the future, to go back to, you might be talking privately, sorry. Oh, yes, sorry. All right. I find silica great for indoors and gives a strong structure, as it does have the outside natural wind for strengthen their branches and structure, with question mark. I didn't understand the question, but there's a question mark. Sorry, mate. Hey, Mick Stones. G'day. Good morning. And just Mick or anybody, if you've got any questions. I think last week with answering questions worked better than me slowing slides, because the slides from my classes this week wasn't directly related to cannabis. It was some disease stuff and things, but it wasn't directly related. So I won't bore you with that. So I thought of the questions worked pretty good last week. If you've had any, I could answer them to my best abilities and show some slides possibly. Just reading the things. I've got a really good question for breeding. Good, green lungs. What is it? Oh, here it is. Can you unfeminize feminized seeds without going to the extent of tissue culture and extraction of the male cells? That can be done in any way. Can you unfeminize a feminized seed? All right. Let's think about that. So if that's got 2 X chromosomes in it, can you turn one of them into a Y chromosome and make it male? Well, good question. That's my head's thinking at the moment. That's a great question. How would I play around with that? Well, if you do with the chromosomes, because if you duplicate the chromosomes, if you go and double them, which is a dihaploidization, which is tissue culture, that's what it's called in that. That's what I'm doing. That will take. It's a direct 100% copy. So if I was to duplicate an X chromosome, it'll go exactly X, or Y, it'll go Y. I can't cross them. How could I get them to cross? Well, the reason why it happens when you feminize in your flower, and when you're inducing it, which is called anthegenesis in flowering, is because you're putting on an ethylene blocker, and the ethylene blocker, it blocks all. You can use any type of silver base, so has your silver nitrite, silver oxide. It is your silver nitrite. Jesus. Anyway, there's also colloidal silver and SDS, your silver diethyl sulfate, to use to that, and that blocks the ethylene that comes out of the plants, and that's what induces the maleness in it. So I don't know. That'd be a question that I'd think about, or I'd be posting that in one of my chat questions on one of my classes. Can you unfeminize feminized seed without going to the extent of tissue culturing and that? Good question, Green Thumbs. I like it. Keep up the good work, mate. So I hope that didn't really help. Sorry about that, but I answered to my best. I don't know is my question, is the short answer. Great question. White ash, g'day, mate. How you going? I hope you're still getting that white ash. Flushing it good. Actually, one of my things to come out soon is all about the white ash. It might be 2 weekends' time. You can self-it green lungs with gibberellic acid. No, that won't work if you put gibberellic acid. Anyway, if you reckon that works, that's good, mate. I hope it does work. I haven't tried it. Not talking about solving it. Make it back, then get a male with a back cross. Oh yeah, cheers, Aussie. G'day, 420 bin. How you going up there in the land of hot lands? Must be pretty hot up there for you at the moment. I was about to type gibberellic acid too. Oh, never heard of that one. I thought it was gibberellic. Sorry, interrupt. How is it? White ashes. Don't know. Because Green Thumbs. Because doing that, I'm pretty sure you can still get herm traits later, can't you? If I'm wrong, well, if you get the herm traits, that's when you backcross it, you inbreed it, and you get rid of all of the unstable codons. So by inbreeding it, when it does it, it goes through recombination and it loses all of its crappy unread codons, which is your gene in the gene pool, where your DNA codons are written in 3 codes, and some of them, they misfire and they're irregular. They got all sorts of types of mutations, induced or spontaneous mutations, and you go and eliminate that by inbreeding it and selfing it. And that's why it increases its homozygosity. And that's what I'm doing at the moment with fruit tingles, because it's unstable, but it's such a freaking keeper pheno. It tastes like fruit tingle lollies in Australia. The fruit tingle, just like it. Like my mouth salivates when I rub the buds. It's a spin out. And then when you smoke it, it tastes the same too. It's just dead set bloody nice, but it's got a herm tendency. It just stresses and whinges, meaning that its heterozygosity limit is really high. So that's how I would do that. So to eliminate the herm traits, you can get rid of them by inbreeding or doing a good. And in my breeding video today, I explained how I did that, how I do it now with epigenetics. Don't do a direct inbreed because you'll get mutant traits expressed, and you don't want any to work with dominant mutant genes because it sucks. Yeah. All right. Next thing, Aussie. Oh, good on you, Aussie Autos. Best educational channel round. Thanks, mate. I try. Need to indeed having lady boys. Don't, Jesus. Thoughts. So why am I talking about completely eliminating the herm from the start? Well, if you want to eliminate the herm, it's because if one of the runs goes a bit bad, and you're never going to have a perfect run, like it's hard to have perfect runs, you'll have there'll be abiotic or biotic stresses, and in those 2 lists there's a shitload. So you'll run into one of those problems, which could be temperature, moisture, bugs, disease, infection, quite a few different things like that, even toxicities, pH, extremes of all sorts of stuff can bring out a tendency for it to herm. So if you've got lower tendencies, which means they're more homozygotic, that means that if one of those stress events comes out, it won't produce antigenesis. It won't go and herm like wind. I'm playing around with a triploid at the moment. Well, I've been for a while, and I put it. It can go under the fan, like right next to the fan, and all that it does is a cool experiment. All it did was it didn't herm at all, didn't produce any anthers, but it grew shit, because you know how if you've got too much wind in front of the stoma where it breathes from, it can't respire properly, and it's going to just whinge because it's too much wind. What's that? Like striglomorphogenesis is when the trees, like at the beach, that's when the wind shape is leaning towards the wind, and it's from a direct relation to the wind. Anyway, I'm off the point there. I'll get back to it, sorry. Green thumbs, do you soak maybe with testosterone? Do you soak maybe with steroids? No. You don't use testo. Jesus. Good experiment, but actually, no, not an experiment. You don't use that. Yeah, like Nexus said underneath, gibberellic acid. GA3 is the correct one. There's 100 odd forms of gibberellic acid, and GA3 is the best one. That's the one I use. And you want to give it what it wants. Well, maybe this might be prompting me, this is down to the last one. It is too, it's the last thing. All right, I'm going to go into detail. Oh, no, it's not. There's some more questions. All right, spewing. Because I could have gone into detail about the seeds. You want 3 things for a seed. It wants oxygen, temperature, and moisture. I nearly forgot there. I think I'm far out. It's so simple. It should be for me because I've been over this 1,000 times. And once you give it those 3 things, that's what it gets it to start. And it depends on its dormancy state to how it's going to initiate. Because if it's in a heavy dormancy or a secondary dormancy, there's quite a few different dormancy states that it can go through. You have to break it out of that. And to break it out of that, that's when you'll usually crack the seed stratification. And you can, so I put my seeds on top of the water, and then because that's going to give them the three things. If you let them sink them or like go to the bottom like I used to do, you're going to miss out on the oxygen factor, because the oxygen is 10,000 times slower to pull it out of the water than it is out of the air through the mass balance transfer equation. And come on, keep going. So by leaving the seeds on top of the water, you can eliminate that and give it its three things that it wants. And then after it does it within about 3 days, you know that you've got a heavy dormancy state in your seed. So that's when you can do some stratification or crack it. So you can, the best way is just to play around with it. If you paid money for the seed, you might be fucked. You want to do this on sort of seeds that's, because you'll stuff it a bit, because seed's small and it only takes a little bit of extra effort, and bang, it's cracked open too much. And then that will ruin its stages that it's got to go through, because it's the seed's a protective layer, and it's inside there. It's got its 2 starch leaves that it's going to form. Then they're white at the start, and then they'll start and grow green and grow cotyledons. They're starch layers that it's got to react with. So by keeping the seed shell on, the seed coat, the tester, it's called, that will guarantee its success. Actually, that'll do. All right, all good. It rattles my brain. Okay. White ash from Purple Bud. Yeah, Nexus. All right. Any more questions? I get what everyone's getting. Geez, I'm a bad reader. I get what everyone's saying with the back cross, but where I'm coming from is to actually physically get a feminized seed and basically turn it reg. Yeah, I understand. It's a hard question. It's a great question. It's playing around with the chromosomes, because that's what it is to do with the sexes. So you want to change those, and you'd have to grow it up, and even by stressing it, you wouldn't induce it and change it. You'd have to and even when you're, yeah, it's just a great question. It's something that I have to think about, and yeah, not sure. I understand your green thumb, green lungs. All right, scrolling down a bit. Any questions, guys? Nearly at the bottom. Okay, so what you're talking about, what I'm talking about. All right. It's hard to include. I should put, all right, I'm down at the bottom. I'll put the link up. Matthew Flick. G'day, Matthew Flick. I'll put the link up if anyone wants to come up and have it see about any. Say anything. G'day, Matthew Flick. I haven't seen you. Say g'day. This is a screen. This is just a thing in the States that you can study. It's at the Cannabis Training University. It's 8 courses that you go through. And for someone who hasn't done much in cannabis, it's a fantastic introduction, because it'll really teach you a shitload. It's videos, mostly videos. There's a few Ed Rosenfeld books, but I learned one thing. If you learn one thing out of anything, it's a bonus. And I learned that about Pikashi. So that was a big bonus for me. So it was really cool. Sort of share that. Love the teachings. Thanks, Green Lungs. Appreciate the nice words, mate. All goodies. What's what I say, Green Lungs? If you could change sex before sprouting the seed, would be a huge time saver. Yes, it would be. That's why I only breed with feminized seed, mate, because I get the same things, and not many people can say they honestly can pheno hunt their male. They know it's good traits, but I can say I pheno hunt my males, which are females, because I use the pollen from I induce it, antigenesis. So I'll go and pheno hunt, and then I'll think, cool. So bang, I've made pollen, which is male bits. And that's how I do it. So I actually pheno hunt both, and then I only get those outcomes, which is the feminized seeds. Pink and orange pistol sugar leaves, frosted like a blizzard. That sounds pretty good. Very good. Nexus. He's got the goodness, he reckons. North. G'day, North. How you going? here up north, I reckon. What do you recommend for cracking the seeds open with? Great question, mate. And it's right down the bottom. So I can go into a bit of question here. Surgical clamps for cracking. Nexus reckons. I use my pliers. So I've used pliers a lot. I got electrical engineering background, and so I can work them pretty good. You don't want it, and you can go too much. So I'll just go to a little crack, or sometimes if it's a really, say if I've got one seed of one strain that I really want, and it's an old seed that I want to get with, I'll go and get my Leatherman knife out. I shouldn't say labels, brand, sorry. I'll get my pocket knife out, and I'll hold it in my hands, and I'll slowly put a slit at the top. And you know there's an opposite end. So you know how the seeds actually open? They open towards the hilum, where it attaches onto the ovary where it's made, not the ovary where it's made, it's made inside of the perigonal bract, but where it attaches to the plant thing, it's inside of the sepal there. It's got this little kink at the bottom, the black little, the crunkle down near that bit. It's the last question. I can go into detail here. All right, I'm going to try and find some studies, and I'm going to show you some seed stuff, because that's what we were talking about. Where is it? Principles of seed. Actually, I'll just go into my plant science and talk about the seeds here, because I think I've got some cool stuff in here to show you. Yes, I have. So let's go in here. So that was the last one. Hey, Stony Creek, how you going, mate? Yeah, so either the pocket knife or the pliers with a real light, gentle touch. And not invite. Well, I'll put that invite thing up now, just in case. And I'm just going to go into detail about the seeds, because seeds are cool. There's the invite thing, but I'm not going to be able to see chat for a bit, because I'm going to show share the screen again, and we're going to talk about some seeds here for a bit, because seeds, it starts with seeds, so everybody's got to get familiar with the good practices of seeds. What's a good thing to start with? Well, seed storage. I really hope that everybody all stores their seeds well. These are some rad seeds. This went through in the video just before about the ripening times. So this is a chart to do the development of the ripening of this cannabis seed. So it just shows the optimum time here to collect, 25 days odd. So if you're going to sort of start pollinating at day 30 or day 40, say, and you're going to harvest at day 60, you're going to get a lot of green and white seeds, and seeds that aren't mature, they're going to be hollow in the middle and stuff. So that's why you've got to do it. And I don't start too early either. If you do it when you first see pistils at day 10, day 12, what happens is it makes the plant slow up. So you want to do it at about day 20 to day 25 is when I pollinate. I'll do my manual pollination. And then the plant powers on, and I get a really good harvest. And from about 4 sepals, I'll get 80 seeds. So I only got to put out sort of 8 bananas or 8 anthers to get those results. Now I better keep going. This is some video stuff. That's how it's ripening. This is how it's actually. This is stuff I'm going to put in video soon. Shit. This is how it starts out. Actually, I don't even want to. Sorry. I was going to do a video for it, but my videos aren't going the best. I'm not quite reaching the people that I want to reach. Yeah, actually. So I'm just going to show it, stuff it, because I probably won't even get around to making that video. Sorry about that. Actually, let's just go to here. Where was I up to? And this is just how it just germinates. So after you get the 3 things, the oxygen, the air, and the temperature correct, it'll start to produce this inside of it. Actually, let's go inside of it first and show how it processes inside, what it does. There it is. So once you imbibe it, and it opens out this way, because this is down here where it connects onto the plant. I can't remember what it was called, but I named it before. But the water comes in and it reacts with breaks the sugars down, and it starts the amylase. So you can mimic that by hydrogen peroxide, H2O2, or iron pyrite, stuff like that mimics amylase. And that starts the process, breaks down the sugars, breaks down the starch into sugars, and then releases the gibberellic acid. And then it starts the formation of its growth development. All right, that's how it starts. And then we come to, so once it's started, then, it's initiated. Gibberellic acid's been with ethylene. This is how to start it. So let's have abscisic acid. So gibberellic acid's down here. And what's this actually? I might confuse you. That's a plus. So yes, it does rupture and weaker. So with gibberellic acid, it comes down as a plus. See, it continues on and it weakens the endosperm, which starts the process. And if you've got C2, that's ethylene. If you've got ethylene, it also helps as well. That's why when you're starting to germinate seeds that are really old, you have to use ethylene. And I use bananas or apples, and pears are another good one that release shitloads of ethylene when they're ripening. And if you want a lot of excess, you cut the fruit and it'll release too much. So and you'll get a triple of response. So you'll see when the seed comes out, if the radical, the white thing, goes hooked, it's and shortened and thickened. That's called a triple response from ethylene. So you'll know that ethylene's working. And then you got abscisic acid, which is, it blocks everything. So actually, who cares about that? That's just really bad. It's just a good thing to induce stress, resistance into your stock. So after it's put out, it's pretty cool. So see, it's got all the fuzzies there. That started all the microbes. So they wouldn't have done this in a lab. This would have been, see, that's grown airborne microbes. That's shit, eh? The Plant Cell Society in Oxford. That's seed germination. I think we pretty much covered it. Oh, cool. Oh, that's to do with the different amounts of stress that you've got. So more abscisic acid will make it more dormant, and the less will germinate it. That's all it cells when it's dividing. It first divides and then it starts to elongate as it processes. Seed dormancy. I'm actually doing a video one was in plan to do for seed dormancy on different techniques to break it. We've sort of done a couple here. They're rad, the sclerids, proteins, seed germination. What's that? Oh, okay, this isn't very good. Disassociation of water. There, right. How the life of the embryo begins. I just explained all of that in the breeding video that I just posted today. This is inside of this, is pretty cool. You see the white starchy bits that you eat that contain all of those goodness that I explained at the start. All of the rad properties, how they're 3 times and 2 to 3 times more beneficial than flax seeds. That's healthy. Okay, very good. That's the first one. Seed maturing. Very good. Oh, and that's just different techniques. That's not really helpful. All right, that's it. That was went into that. And it's been, holy hell, 40 minutes already. I'll scroll up, see if there's any more questions, but I reckon that's it for today. That's right, I was up where Stony Creek was. Berries dipped in my hash pipes. There's someone. Look, Nexus is back screen. Sorry.
Yeah, g'day, mate.
G'day, Nexus. How you going? I'm going to scroll up. How are you today?
Yeah, really, really whacked. But yeah, doing better, mate. Yeah.
That's good. Do you have any questions or anything that you can add or?
I should say use gibberellic acid.
Yeah.
It will help in that. It will help in that process. You'd have to design an experiment. You'd have to use ethanol as well.
Yeah.
Yeah.
Here's one from Jim. Hello to Jimu Sama. How are you? I hope you're doing well. You just use skins to avoid cutting the fruit. Yeah, you can use the skins. I haven't got one of my ones is an ethylene meter, and I haven't got that yet. So when I get that ethylene meter, I can confirm on how much ethylene they'll do. They'll probably release a heap until they'll dry out. So that's my guess. What do you think, Nexus?
A tomato will give you half the amount of ethylene as a banana. So use a banana skin and half a tomato if you want to really boost it. If you want to do it quick, if you're in a rush and you've got 12 hours, that's what I would do. But that's kind of forcing it a bit. It might be too much.
East Jimmy says, thanks. Yeah, was on ethylene use in bananas. Yes.
Yeah. Yes.
Well, they're the three things I've got. I can show you that, I suppose. This is the.
Right.
Yeah, there's the. Where is
They
it?
give you ethylene when they're still a fruit, and you can break them down into potassium.
Here you go. Shouldn't be
They
showing.
make great potassium.
Yes, you have to break them down first.
Yes.
But yes, they sure as hell do.
Yes, 10 kilos of bananas will give you about 2 kilos of potassium, thereabouts, if you do it right. A lot of heat, a lot of times, but you eventually get it. Can you guys hear me all right? I'm using these little Galaxy buds with my computer. I've got them.
Yeah, you sound pretty good. To me,
Sure.
you do.
Sweet.
I'm just trying to go through. What was that one? I've got a good meter. I've got a good chart because I'm putting together a lot of dormancy stuff at the moment. Well, I was actually before my study started up again.
Subject a little bit. That's that scary shit.
Pardon?
That's that purple stuff. That's the 33.7%. It's just God.
What is that, a bud?
Yeah. Yeah, mate. That's the end of the bud. Yeah, that's what I got left.
Hang on, I'm trying to spotlight you. No, wrong person. Hang on. I don't know how to spotlight you. I've seen other people do it. No, that's me. Not me.
You click on my picture and there's the 3 little dots in the corner. And you click on the 3 little dots and it'll go full screen.
3 little dots. Edit name, edit mic, kick from studio, and ban from studio. No, don't want to push any of them. Oh, that one. Remove solo layout. Solo layout?
That's the one.
There you go.
There you go.
What are we looking at?
What are you looking at?
Put your hand behind it.
Put my hand behind it. All right. Put that down.
G'day Blinky, how you going, Aussie Jeffro? G'day Dan Boy Grows.
It won't really help because this camera's got like fixed focal.
All we can see, it just looks like it's white and frosty as hell. What is it? What bud is it?
Dosey Doe crossed with Granddaddy Pert, and there's a Larry OG in there in the mix too. So it's a really potent berry. Oh mate, the smell is just amazing.
Well,
amazing. It's very fresh. It's very fresh. Yeah, coming to a Canatrek can near you sometime soon, I think.
Yeah. Very good. Well, I can't find that seed sprouting and chance. Oh yeah, here it is. It's under seed sprouting. I was looking under I was making a seed sprouting video and a seed dormancy video. So I reckon here it is. Here, I think. Is that the chart that I want to show you?
Well, the techniques for both would be relevant. Yeah.
Ethylene biosynthesis.
That's a really good question, Corey. Unfeminizing a feminized seed, because I think feminized seeds are the curse of the society.
What was that last bit? You think feminized seed what?
Feminized seeds are degrading some of the strains, I think.
Let's talk about this. I'm not going to show that. How do you think?
Like I see all this stuff coming out as feminized seed, and it should be testing way higher than it actually is after it's been grown. The stuff that's like, they say it's going to give you 25, 26%. It's not. It's coming out 16s, 18s. And the only common denominator I can find is all the stuff that's going from feminized seed.
Have you tested? Is that from one grower, or is this across the board? Because mine tests fine.
No, this is from about 12 different growers. I know I know one guy that grows for Beacon. I know 3 people that work for Canatrack. I know a guy that works for another company, but I can't remember the name. It's some weird name. But yeah, I know a bunch of different people. They're having a consistent problem with potency with feminized seeds.
Hmm.
And these are
12.
professional.
That's a high call, 12, because I know quite a few people that actually, that's why this is a good topic to discuss, because I got some friends in BC who do it, and they test their stuff each grow, and theirs is fine.
Yeah, I think the problem is where these people are sourcing their genetics from.
Oh, there you go. Yeah.
I think that's the issue. It's legal
Definitely.
stock. It's coming in. Well, it's coming in from the United States. Yeah,
Well,
and
if
like,
you're getting stuff that's got mutants in it, you're breeding with mutant genetics. And you remember, there's heaps of ways to induce spontaneous mutations. And great one is to get them X-rayed. Let them go through the borders that come through, and let them get scanned. And that denatures DNA and induces mutations. And if you're breeding, it's like breeding with handicapped people. It won't work. So, yeah, now I understand why they're getting those results. Mine isn't anything like that. Mine is all stable, so the genetics are still the same from when they were bred from, and I haven't had any problems with it. I suppose I don't sort out Australian genetics to breed with for that reason, actually, because I've found the mutations are so high. That's why we've got Australian bastard weed, world renowned. How you going, Raymond Cowell? So yeah, that's cool. And to eliminate the mutant traits, you have to breed 7 times back through them to get rid of them, and that's a task in itself. And I don't know really anybody who's going to put that much work into one strain that they love it that much.
I did. I backbred Australian. I started it in 2014, and the project finished in 2020. I spent 6 years.
Why did you work on that for so long?
We ended up backbreeding it. It got up to 90 times. We back-crossed it with itself 90 times.
That was the one that you went back. It went back thousands of lineage years.
Yeah.
Generation years from its
generation. Yeah.
Yeah, from its back crossing with its parent.
Yeah.
And that's pretty cool.
Well, the resulting hybrid was a consistent 31% THC all the time. Just didn't matter how you grew it. It just had that. If you gave it really intense light, it just got really dense. And yeah, it was enormous. But for some reason, they stopped work on that project because they found something better. So all that work.
Well, it'd be pretty hard you'd have to grow it out because you're not growing feminized seed. So they have to grow out, do a pheno hunt, and then make sure that they're getting the females, then breed with those ones back with the parents. So it would have been a very tedious task. So I can probably see why they stopped.
Well, yeah, it was tedious, man. Yeah, it gets old after a while. Yeah.
Cool. Well, is there any more questions, anybody? Because I'm going to wrap it up, I reckon. I appreciate everybody for coming in. And I'll be back here next Saturday to share what I've found. And I know that next week I'll be talking about minerals in particular, elements, particular the 19 elements that constitute and what they do. For instance, like today I talked about silica and what it does and how it's important with its with its stresses and its osmotic protection that it helps. So yeah, that's something that I'll be talking about because I know that in my soil class next week they're going into heaps detail on the elements itself and why each element is important separately for the plant. So that's going to be pretty cool. So yeah, that's all I can suggest. Thanks everybody for turning up again. G'day Buttercup, we just rocked up. Thank you everybody. And next week's video. Beg your pardon?
No, I was just saying g'day to Aussie Jeff in the chat, mate. A couple of people I haven't seen for a while.
And next week's video that I'm going to put up will be, I think it's the, shit. Oh, bear with me then, I'll tell you. I'll give you this a little sneak, whatever you call it, inside information.
Preview of next week. Yeah.
Preview, that's the word I wanted. Thank you. Oh yeah, it's fungus-resistant plant genetics. So there's an 8-minute and 30 video on installing and testing fungus-resistant genetics and systemic acquired resistance, induced acquired resistance, and I go into a bit of detail. I can show you the photo actually. Get you all G'd up. Where's the photo? I'll just pull it up. View on YouTube. I've only got a few of these videos left too. I'm not making any more at the moment. Sorry. I'll just share the screen so I'll show you. I'm just not quite getting the views that I thought I would, and it takes a lot of effort to put into this. And I'm doing my studies at the moment too. There it is, fungus-resistant genetics.
G'day. This is a video on...
No, I'm not playing it.
Yeah, there's a preview of next week.
It's a video on fungal-resistant genetics and systemic...
No, I just wanted to show you. But yeah, it was... Sorry, that didn't go too well. Yes. G'day, Blinky. Come in. Much appreciated. Good on you, Lene. Thanks for the nice words. All right, well... Nexus, do you have any things you'd like to say before any notes or anything? Goodbye to anybody?
Man, I'd just like to say thanks to the 420 community for being so supportive while I've been having a bit of a battle. So just thanks to the people that helped out.
Good on you. Good on your 420 community. Oh, my tap's just become unfrozen.
Oh, good. There's running water.
Just come back on. I've got to turn it off. Hang on. It was minus.
So it's that cold down near your pipe's freezing up, right?
It was -6 last night, so it was a bit chilly.
Good. Well, it got to -2 here the other morning, so yeah, yay, climate change.
It's 21 Fahrenheit.
I'm not kidding, mate. -2 in Queensland. Yeah, it's fucking crazy. And it's sunny day and it's 12 degrees. It's climate change is real, kids.
Yep, it's full on. Well, thanks everybody for tuning in. There's 16 people watching. Good on you. I appreciate your questions to hold the show open and your turning up to keep the show alive. So it's good on you. So next week I'll discuss that stuff and keep some questions for next week, please. And appreciate it again. Good on you, Wombat Organics. Just turning up. Well, not turning up, sorry, but I just want to acknowledge you. And North. Thanks everybody. Cheers. Pardon?
Oh, the politics around climate change is fake, not the actual thing. But anyway, all good.
Climate change is fake. No, it's not fake. It's bloody real. He's just joking. We're all a sarcastic bunch in Australia too much. That's why Wombat's it's not real, is it? Pig's ass.
No, it's not real. It's not real. It's -2 in Queensland.
What do scientists know? They're full of shit. They don't do research and stuff every day of their life. They just know crap.
The really smart scientists are really high because we know what's coming.
Yes, look out. That Great Barrier Reef blooming told us that about 15 years ago. So
they told us that in 1996 when ICI Chemicals accidentally, on purpose, built 1 million tons of sodium cyanide that poisoned the reef. That, 5 years later, they mined it for all the lime in it.
There you go.
Same company that's poisoned it, mined it. Oh, by design? Yeah.
Yeah.
We'll say that for some other time.
All right. Cheers, everyone. Thanks again. See you next week. And bye-bye. Happy breeding, happy growing to you, and have a very good day. Bye.